Since we are talking about probabilities, as we need to deal with probabilities when we are doing finding like genes or promoters, it's important to know about some probability distributions. So one of them, which is most frequently used, is binomial distribution. So how can we define a distribution, a probability distribution? So a probability distribution is a table or an equation that links each outcome of a statistical experiment with its probability of occurrence. Whereas a binomial distribution is a probability distribution for independent events with discrete outcomes. So you have independent events and their outcomes are discrete. So either you can have yes or no situation, ones or zero, you can have head or tail. So these kind of situations where you can apply these binomial distributions. So it is used when there are exactly two mutually exclusive outcomes. So one more thing is that these events, they are mutually exclusive. So either you can get head or you can get a tail. So everything is associated with its probability. So for example, if the coin is fair, you can get 50% head and you can get 50% tail. So that is a situation where you can expect one of two outcomes with having some underlying probability. Here are some assumptions which are important as far as this binomial distribution is concerned. Number one is that there are n identical trials. Trials means experiments. So for example, if you want to toss the coin 10 times, then your n is equal to 10. Each trial results in one of the two outcomes. So either you get head or tail in each trial. The probability of success is P. We mostly use P to denote the probability of success. Obviously for failure we can use Q. The trials are independent, that is the outcome of one trial does not influence the outcome of any other trial. So having tail on one of the trial does not influence the outcome of the next trial. So they are kind of independent events. And the binomial random variable S is defined as the success. So if you are interested in, for example, 4 tails, your S is equal to 4. So S is equal to 4 out of n, and that is equal to 10. So we can use these n, S and P in one of the equation which is provided as probability mass function. So here in the S function is most of the time we use in these even biological situations. So as per this function, we can get the probability of successes. So these successes can be anything between 0 to 10. So if you are throwing this coin 10 times, your n is equal to 10. So you can have 0 heads and you can have up to 10 heads. So in between you can calculate the probability of any number of heads. So if you are interested in 4 heads, your S is equal to 4. So we can calculate the probability of 4 heads with this equation. This middle part in this equation, this part where you can calculate the combination. So how many ways you can expect that success. So you can have 4 heads, 4 consecutive heads. You can have head on the second trial and then third and then sixth and 10th, so and so on. So how many possible ways you can see or you can find that success. So that is defined in this part. We open up this thing into a factorial equation which is coming in the next page, whereas in this equation n is number of trials, S is success, P is the probability of success, and 1 minus P is the probability of failures. So sometimes we replace 1 minus P with Q also. Maybe we can have a simplified version of this equation. So here is the equation. This S over here is how many successes you are interested in, and here is if you are interested in, for example, 4 successes, there are 6 failures, so that we can get it from n minus S, which is right here. This combinations we can calculate, as I said, with this formula. So here you have n into S, which breaks up into n factorial, n minus S factorial into S factorial. We can have an example of a factorial. So factorial of any number is the multiplication of that number into its smaller number than this and goes up till 1. So if for 4 factorial, you can have 4 into 3 into 2 into 1. So its answer is, in this case, is 24. As I said that we can simplify this equation. So here we can break that factorial part. We can put it there, and we can also have this Q rather than 1 minus p in the equation. So there are different forms in which you would see this equation in the literature. Let's do one example. So example is, what is the probability of seeing 4 Cs in a sequence of 10 nucleotides, not necessarily consecutive? Here is the probabilities which are given. So it's a fair trial, guys. So we expect 1 by 4 for each of them. So for each of A, T, G or C at any location, we expect their probability is 0.25 or 1 by 4. So here the die is fair. So most of the cases it's not. So sometimes, as we have seen earlier, that nature places some nucleotides most often at some places. Success here is seeing a C, and here we are interested in 4 successes. So S is equal to 4, P is equal to 0.25, and N is equal to 10. Let's put it into this equation. So here we solve it using this equation. So we have P of S is equal to 4. So this is given by n factorial divided by s factorial into n minus s factorial into P of S 1 minus P n minus S. When you plug in the values, you have 10 factorial divided by 4 factorial and then 6 factorial, and you have the probability of success, that is 0.25. So we have raised to power 4, whereas the remaining guys are probability of failures is, then since we have we can expect any one of them like to be there. So each other 3 nucleotides have a probability of 0.25. So if you look with reference to this C, not seeing the C is actually the frequency of or the probability is actually 0.75. So we put that 0.75 over there. If you are not seeing C, that is your failure. You can see either T, G or A, and their probabilities, they can then add up. So when we solve this equation, so our answer is 0.1459. So it's around 14.5% probability of seeing 4 Cs in a sequence of 10 nucleotides. Here is another question or example which I can solve with you guys. Sequencing reaction is performed with an error rate of 10%. What is the probability of observing a correct nucleotide in 2 out of 3 experiments? So guys, this error rate is a success here or a failure here. Look what into you guys are interested in. So you are interested in correct nucleotides. So you want to see what is the probability of seeing those correct nucleotides. In that case, that is your success. Seeing an error is your failure. So here we can plug in the values. We can have P is equal to 0.9. We can have Q is equal to 0.1. And what is N here? How many experiments you want to do this? For 3 experiments. And how many correct nucleotides you are interested in? So that is your success, S. So you are interested in S is equal to 2. So let's solve this factorial part first. So we can have how many combinations, how many ways we can see these 2 successes. So we plug into that equation. When we solve it, so it is n factorial, which is actually 3 factorial, divided by s factorial, which is 2 factorial, and n minus s factorial, 3 minus 2 factorial. When you solve it, is 3 into 2 into 1, which is 6. Then 2 factorial is 2 into 1, which is 2. And then 1 factorial is obviously 1, so that is there. So there are 3 ways you can see the experiments in which you get the correct nucleotides or correct base calls. It may be like this. So you can have a success, success, and a failure. You can have a failure and 2 successes, and you can have a success, failure, and success. So 3 ways you can get your these 2 successes. So we solve this part. Let's move to the remaining equation. So here we put everything in the formula. So probability of seeing S is equal to 2. We solve it this part, so that is 3. And then for P, which is the success probability of success, and then P raised to power S, which is probability of success, that was P, and is for S is equal to 2 in this case. And then for Q, it is obviously N minus S, so it goes to 1. So when we put it there, so it's 3 into P, which is actually your 0.9. square into q, which is 0.1. So when you solve it, you get to 0.243. So it's kind of 24.3% is the probability of your success, is that two of your experiments, in two of your experiments, you will observe a correct nucleotide. How do these binomial distributions they look like? They look like normal distributions or like bell-shaped curve if your p is equal to 0.5, or otherwise they are skewed. So if your probability of success and failure is equal 50-50, or if for example you have a fair coin and you are tossing it many times, you can get this distribution like it looks like this. Otherwise, wherever your probability of successes is, it will be skewed as per that probability. For every distribution, we need a mean and a standard deviation in order to do some test statistics. So mean of this binomial distribution is given by n into p, number of trials into probability of success, and standard deviation is np into 1 minus p square root. Remember, we take variance first, and then we take the square root of the variance, so that gives us standard deviation. So far we have observed that binomial distribution is used when there are two mutually exclusive outcomes for an experiment.